Hey guys, Dr. McDonald again. It's time for us to finish up chapter 3 in this part 4. Let's see if I move myself a little bit. Molecular structure of DNA. This is where we left off. We have talked about this, but there is kind of a lot to it. So let's look at this. Now all the DNA are made up of nucleotides. There's only 4. There's adenine and thymine, and those guys bond together, hydrogen bond together between the 2 strands. If you look right here, you can see thymine and you see adenine. Now these are on 2 different strings, 2 different ones. These guys hydrogen bond and they kind of stick together. And then we have cytosine and guanine, and again, if you look, you'll see these guys hydrogen bond. They actually do 3, and these do 2. So those are our 4 nucleotides. Now let's talk about this guy. An individual nucleotide is going to have a phosphate, PO4. You can see it there, and it's got a -2 charge. You got the phosphate double bonded to an oxygen and single bonded to 3 other oxygens. And if you look, you'll see a sugar. Now, we kind of hit on this when we were doing carbs in the chemistry chapter, but this guy, this guy would be hydrogen, would be deoxyribose. How do you know that? Because right here, I don't know if you guys can see this thing, it's kind of annoying, but right here on the end of it, it should be an OH if it's ribonucleic acid, ribose. Deoxyribose has a hydrogen. The hydrogen is implied there, but it only has 1 hydroxyl group, so I know this is DNA, but it's that 5-sided sugar. So the phosphate bonds to the 5-sided sugar, and it bonds to what we call the nitrogenous base, and that's the thing that differentiates different nucleotides. You look over there, thymine is a single ring, adenine is a double ring, guanine is a double ring, and cytosine is a single ring. So that's how these 2 strands of DNA are held together with hydrogen bonds. The hydrogen bonds are also going to cause it to twirl, making that famous double helix. Now, that's okay for this guy, but now we're going to talk about one of the more important parts. I've talked about how the 2 strands are kind of bound together, but what binds a nucleotide in each strand? And this is it, guys. Now, you don't need to know a whole lot about this because it's kind of, technical. But there are 5 carbons in a 5-sided sugar, duh. So there's carbon number 1 right there, carbon number 2, carbon number 3, carbon number 4. Carbon number 5 is actually off the ring because we have an oxygen in one of those spaces. Carbon number 5 is the one that bonds to phosphate. So we call that the 5' end. It's just 5 with an apostrophe. If you look right there, you can see that this is the 5' end because that's a phosphate. And on the other strand, you can see that's the 5' because it's the phosphate. So we have a 5' end. Now, this guy, his number 3 carbon is going to bind to the next phosphate in the line. So if this sugar number 3 again is going to be, let's see, 1, 2, 3 right there. This guy will bond to the phosphate of the next nucleotide. And that bond, dehydration synthesis, of course, but that bond will, because remember there's an OH right there, and we're going to, now this, in this case, it's kind of funky, but we're going to bond, that OH is going to go away, and it's going to bond to that phosphate. But in any case, let's see if I got that right. 1, 2, 3. Yeah, because that's number 4. The reason I'm going through all this trouble, guys, is it's really important that we know the 5' end and the 3' end. And something you may have already noticed, that on one strand, and this is one strand and this is the opposite, the 5' end and on the 2 strands are going to be on opposite. So if this is 5, then the one right across is going to be 3. That's called anti-parallel. That's just the way DNA bonds and the way it comes together. So the 5' end of one string, and you can see it right there, one strand, and there's the 3' of that same strand. These guys are running opposite directions in the sense that, you know, the sugars and the phosphates, the way they bond. Okay, a lot to that. Okay, guys, this is codons. So let's look at it like this. DNA is a language that has exactly 4 letters, A, T, C, and G. Now, these 4 letters can make words, and these words are always 3 letters long, and that's what a codon is. Codons tell the ribosome which amino acid to put next. That's it. So if you look right there, you see UUU and UUC codes for phenylalanine. Now that UUU, remember that in RNA we don't use thymine, we use uracil. So anytime there's an A on the DNA, instead of putting a T on the RNA, we'll put a U. That's all it means. You notice there's 2 for phenylalanine, 2 for leucine, well actually 6 for leucine. So all of these guys have different codes. In some cases 4, 6 of them code for the same thing, because we've only got 20 amino acids, but we've got 64 combinations of 3 letters to make words. So the only ones I really want you guys to have down pat are 4 of them. AUG. AUG is the abbreviation for August. But what that means, of course, is AUG codes for a specific amino acid called methionine, and that is always the first amino acid in every protein, whether it's people or even bacteria. Bacteria have a variant on methionine, but basically, yeah. So AUG is the start codon. You got to have that in order to have a protein. So this guy just keeps going, but how does it know to stop? Okay, that's enough. We're finished. Well, this is what these guys for, these 3. UGA, University of Georgia, UAA, and UAG. These guys are all stop codons. So when the ribosome comes to one of these guys, it stops. It's done. It's over. That's the stop signal. So I want you to know the start codon and the 3 stop codons. I want you to understand about the 3-letter words and all that, and what they actually mean. They just code for an amino acid. Tells a ribosome which one to go next. Okay, let's go to the next one. Got to move me again. Okay, genes. What is a gene? A gene is a part of the DNA that has the recipe for a protein. That's it. Not all the DNA. As a matter of fact, most of the DNA is actually just spacers. But a gene is a part of the DNA that gives you the recipe or the sequence of amino acids, and that's what makes the different proteins. We already talked about the 4 letters making up 3-letter words, those codons, they call them triplets. So here we see the DNA. They call this the template strand. This is the one that we're going to actually copy. But the copy we get is going to look like this one, except for the U's and the T's being changed. But the template strand is the one that we're going to copy for RNA, and you can see there's TAC, which is going to be the AUG codon, the start codon. And then you see and it reads it 3 at a time. And I'll show you how and why and all that good stuff in just a minute, guys. But you can see it goes all the way through and it comes up with all this sequence of amino acids, which are so critical, okay? A lot of diseases are from having bad sequences in there. Okay, let me move me up again a little bit. RNA. So we talked about we made an RNA copy of the DNA for protein, but actually there are 3 kinds of RNA. There's more than 3 kinds, guys. There's like 5, but we focus on 3. We have one that's called messenger RNA, mRNA. We saw that. That was the copy we made of the DNA. Let me go back and just show you. That is messenger RNA. mRNA is the way we abbreviate it. Then we have ribosomal RNA, and that just means that inside the nucleolus, we already talked about, we make the ribosomes, which is partially made of RNA and partially made of protein. And the part of the ribosome that's made out of RNA is called ribosomal RNA, which kind of makes sense. So you just abbreviate that rRNA. Now the last kind that we really want to focus on is tRNA, you could call it transport RNA if you like, because it's really going to be the one that actually gets the amino acids and brings it to the ribosomes. We're going to see how this all comes together in just a minute, guys. But I got to tell you about the little pieces first before we go too deeply in this. Okay, transcription. Transcription is when we make a copy of something. You know, if you go into a courtroom, you've got court reporters, and used to they would type on a little machine. They would actually take down what everyone was saying and transcribe it from spoken language to another code, and then eventually into a typewritten or written language. So what this is doing, transcription, is where we take the DNA, the coding part, the gene, and we make a copy of it. We make an RNA copy of DNA. Okay, this starts in 3 places. It's kind of silly. It's so obvious. You got a start, you got the middle part, and you got the end. Okay, we call the start initiation. There is a part on the gene that has a... Ma'am? I can't talk right now. Hold on, guys. Let me see. Guys, I am so sorry about that interruption. But in any case, stage 1, the initiation, there's a certain part of the gene called the promoter region, and it's kind of the signal for the enzymes that make the copies to, this is where to start. It's like a start here thing. Elongation, there's that enzyme is called RNA polymerase. It's going to take those 2 twisted double helix, and first thing it's going to do is untwist it. And you know, those guys, those 2 strands are just held by hydrogen bonds. It's going to actually break them. I mean, it makes sense. If I'm going to make an RNA copy of just one strand, I have to break them apart. The coding strand, the template strand, is the one that we copy. And this guy is going to... Now, let me just give you this little overview. if you put A and T and C and G in a proximity to each other, they're going to come and bond, do the hydrogen bonding. It takes nothing. What the RNA polymerase does is bind one nucleotide to another on the same strand. That's the work that we have to do. That dehydration synthesis. RNA polymerase is the guy that does it for this. He's going to make sure that we get the correct one, the ACG or U, that goes with what's on the DNA. It just keeps going until it gets to the end. Again, there are the UAA, UAG, UGA is a stop signal, and it stops. And then the mRNA is done, and it goes out the nucleus to go to the ribosomes. Okay, I'm in my way again. Transfer DNA, tDNA, I'm sorry. Okay, transcription, we get the DNA, sequence of nucleotides, and we make a copy, but it's actually we call it the complement. So if it's A here, it's going to be U there. If it's T there, it's going to be A. So that sort of thing. Transcription factor is going to, remember the DNA is wound around those 8 histones. It's got to loosen it from that, binds to that promoter we talked about, and then it's going to start with the transcribing by making a copy. The transcription factor is also going to help the RNA polymerase to bind to that guy. So RNA polymerase, we've really kind of talked about this already. It's just really repetition. That's it. That's it. It's just basically what we've already said. Now here's a good picture. Okay, so there's the DNA, there's the coding strand. If you look right there, there's that promoter region, which is where the RNA polymerase, let me pull me down so you guys can see. That's where the RNA polymerase is going to start, and it's just going to go till it gets to that stop signal, the termination. Initiation starts, elongation, it just keeps making that copy, that RNA copy, till it gets there. Okay, now here's something. RNA polymerase always starts on the 5' end. Here's another little piece of information that's going to be really good for you. Everything that we do with DNA starts 5 to 3. We never go the other direction. We always start on the phosphate end of the DNA strand. That's just the way it is. So, I'm sorry, phosphate side of the RNA. When we start making the RNA, we start 5, which means we start on the 3' end. Okay, don't want to confuse anybody. Let's go back and think about this. I said that the 2 strands of DNA, if one goes 5 to 3 like this, then the other one will go 3 to 5 like that. They're in opposite. So if I start the RNA on the 5', that means I have to start on the 3' end of the DNA. Hopefully that makes sense to you guys. Okay. So and of course, after this RNA polymerase has gone past a certain part of the DNA, it actually comes back together and rewinds. We only keep the section unwound that we're actually working with. So there's that mRNA. You can see the nucleotides coming naturally going to the one that they're mated with. And then the RNA polymerase actually does that synthesis that will kind of tie the string of nucleotides together. This not too awfully difficult. Transcription, we got some others that are going to be a little bit more challenging, but transcription's pretty straightforward, guys. Let's continue. I'm going to try not to run you over 30 minutes, but I really want to get finished with this in this video. So anyway, the DNA, RNA polymerase, promoter termination. This is basically the same as that other one. This is just kind of broken down, but it's a little bit larger, so maybe it'll help you see. Okay, I got to move me again. Translation. Okay, guys, this is going to be a little bit more difficult. Translation is where we take the RNA, the mRNA, and we turn it into a protein or a polypeptide. Translation, if I were to get someone who spoke Greek to take my videos and record to dub in the Greek language, that would be translating. They would translate what I'm saying in English language into the Greek language. So we're translating a series of nucleotides, a sequence, into a sequence of amino acids. That's what translation is. This takes place at the ribosome. All three kinds of the RNA are involved. Obviously, the rRNA is going to be there as part of the ribosome. mRNA tells us which one goes next. tRNA goes and fetches these guys for us. Okay, I can come down a little bit. Okay, here's the sequence, guys. The mRNA, the ribosome, if you guys remember, have two subunits, a small and a large. They're actually separated until they come together to make a protein. The mRNA is going to attach to the small subunit, and the large unit's going to attach to it after this, and the mRNA is going, of course, the start codon, the AUG, is going to go in first. Now here's something, it's anticodon. So I've got AUG as the start codon. What is the sequence? What is the sequence on the tRNA that matches AUG? Well, A, we got a U. U, we got an A. And G, we got a C. So the anticodon is what's on the bottom of the tRNA. So I've got the codon, the AUG, and then the anticodon is on the bottom of the tRNA, and it's going to be the part that matches the codon. And the tRNA that has that sequence, it happens to be the one that carries methionine. That's how we get the right amino acid that matches the codon. I'll show you a picture of the tRNA and it makes a little bit more sense. As soon as he drops his off, and he's going to move after it attaches to the protein chain, and another tRNA is coming in, and it's going to just keep repeating until they get to the stop codon. Okay. Protein synthesis, there's the codons. The mRNA is going to have the complement. Now here's the thing, guys. You remember I told you that the codon that's on the gene, we're going to actually have the complement to it. Well, guess what? The anticodon on the tRNA is going to be the same as the coding on the DNA. So you see TAC there, UAC is what's going to be on the tRNA. Why is that? Because that's what matches AUG. That's just as simple as that. So there's the tRNA. It kind of looks kind of boxy, kind of like a plus sign. But there's the methionine attached to it. So this guy is going to go, and he's only going to bind to the guy that matches him, which is the AUG. And then the next one comes in, and he's got proline, serine. All of these guys are going to match the codon. These are the anticodons. And then there's that UGA, which is going to be the stop. We don't have an anticodon for that, because when we get to that, the ribosome comes apart and it's done. Here's another view of it. Now, on the large subunit, we've got 3 special areas. These areas, the first one is the A, the second one in the middle is the P, and the third and final one is the E. If you need a mnemonic, think about King Kong, APE, ape. The A subunit, this is where the AUG first comes. Not a whole lot happens there. It actually, the ribosome moves, you know which direction it moves to? 5 to 3. How about that? Is going to move, and the AUG is going into the P. Now that's where the tRNA comes. He's got his little amino acid, and it's at that point that the ribosome will take the previous amino acid and this one and do the dehydration synthesis and link them together. Now, if that's the first one, they're just going to hold on to that until the second one comes. Okay, when this guy has done that, the ribosome is going to move to the E site. The E site as in exit. This is where the tRNA is going to be released, and he's going to go and get another amino acid. Now, this guy's not going to release until you have another one in the P, because we always are going to have a tRNA that's holding the whole string until we get to the stop codon, and then we're just going to, you know, let it go. Okay. Let me move myself up. There's the transcription. We talked about that. There it is going through the nuclear pore. There's an amino acid. There's, don't worry about that one. There's the tRNA, presumably that matches this, and there's his anticodon. And here again, we've got a bigger thing. So this is the 5' end, and this is the 3' end. So again, we always come into the A site, and then the ribosome moves, and so we slide down 3 codons, or three nucleotides to the P site. That way we keep the same word. And then that's where we actually do our business of synthesizing the bond between the previous amino acid and the one that the tRNA's got. Once that's accomplished, the ribosome moves again. This guy moves over to the E site, and as soon as the next one comes into the P and gets bonded to the previous one, then this guy is released and goes and fetches another one. You guys can look at this. This is really something that it helps to watch a video. You can see this in action. It's kind of hard to explain this, but hopefully I'm getting some of it. So this is the whole sequence: transcription and then translation. Don't mix those two up. Think about, I'm changing from nucleotides to amino acids, and it's entirely different. So I'm translating. Transcription, I'm using the same language. I'm just, you know, using a different code, RNA instead of DNA. The cell cycle, we talked about this in the lab, and I'm sure you guys are not in my lab. You've talked about it in your lab. Cell cycle is the method that the cell goes through to reproduce itself, because cells get old and we also have growth. You know, a baby has to grow a lot of new muscle and bone and all kinds of tissues to increase from newborn size to at 1 year of age. So the cell cycle, we have this humongously long phase. We call it interphase. That's just the cell doing its normal job. If it's a liver cell, it's producing bile and, detoxifying things and making proteins. It's just doing its normal thing. When it gets the signal to divide, then it goes through mitosis. Mitosis is not cell division total, it's just the division of the DNA. And again, so here's the interphase. Now this phase can be as long as years or as short as days, depending on which cells and which part of the body. And there are some cells that don't divide at all. Those guys they're in something called G0, and that's like the neurons. They don't divide. You don't get any more brain cells or nerve cells anywhere. Okay, but if it does have the thing, G1 is where we're going to be growing. We're going to copy our centrioles. We're going to replicate all of our organelles. I mean, if we're going to have two separate cells, then we have to make sure they're all prepared, that everybody's got a full list of equipment. Now, G1, we have a G1 checkpoint. The G1 checkpoint is kind of like proofreading to make sure that everything is on the up and up. We're going to check for errors. If we pass that, then we go to the S phase. The S phase, we continue to grow, but this is where we copy our DNA. We make a DNA copy of DNA instead of an RNA copy like for transcription. Then we're going to go into the final growth spurt, and we're going to have a checkpoint right here, the G2 checkpoint. This is a final check. We're also making sure our DNA was copied correctly. If the DNA is not copied correctly, it can go kind of wild and divide without any rhyme or reason, and that's what we call a cancer. But if it does pass that, then we're going to go into the phases of mitosis. Now, mitosis is just this guy right here: prophase, metaphase, anaphase, and telophase. The entire cell division, though, is also going to include something called cytokinesis, not a part of mitosis. It actually starts in anaphase and kind of ends after mitosis is over. Okay, interphase. The big thing in interphase is, well, we've talked about those subphases, and they also put the G0 in there. The nucleus, the DNA is going to be chromatin. And if you remember, I said chromatin is just kind of spread out DNA. It's there because you have to have it loose in order to get to it to make a copy. But when we get ready to divide, moving that loose stuff is not very practical, and it would cause breakages and all kinds of nasty stuff. So when we get ready to divide, we're going to take our chromosomes or our genes, we're going to wind them up. We're going to twist them and put them in a very compact package so that they can be moved without damage. Okay, let me get out of the way. Okay. We make a copy of it. Now, this is a chromosome, but when it's bound together, we call it a chromatid. This is an exact copy of it. Now, this red one may be my chromosome number 5 from my mother. The blue one is chromosome number 5 from my dad. There's 4 copies of the chromosomes. Normally, I have 2 chromosomes. Of the 23 chromosomes, I have 1 copy from mom and 1 copy from dad, and then the sex chromosomes X and Y, depending on your whether you're male or female. But this is what we do. We make a copy of this right there. We also got to make a copy of dad's. So when the two cells start off, they're all going to have the right number of chromosomes. Not having the right number of chromosomes is usually lethal to the baby. Okay. Or, excuse me, we're not talking about meiosis. It's usually lethal for the cell, and it can cause all kinds of problems. Okay. Now, this guy is called a kinetochore, and basically what it does, if you got one on each of those strands, and it basically holds them together, eventually we're going to break that apart and move those individual chromatids to opposite sides of the cell. But this is the way it goes. And that's that X shape that, most folks associate with DNA, but it actually only does that for a very short time. Okay, here's some of our phases. There's our prophase. Prometaphase, also called late prophase. It's just part of it. Now, prophase, how can I tell when prophase? Well, I've got those centrioles, and they've moved to opposite sides. The nucleolus has gone away, and the main thing I'm going to look at, I no longer have chromatin. I've got actual chromosomes that I can see through the microscope. So that's letting me know I'm in prophase. Metaphase, how do I know that? I see all those chromosomes lined up in the middle of the cell, just like that. In anaphase, that's when those chromosomes that are connected by the kinetochores are pulled apart, and then they go to opposite sides of the cell. When they do that, they make almost like a V-shaped. Telophase, they actually get to the opposite sides of the cell, and they start forming a new nuclear envelope, because the nuclear envelope's got to disappear because you can't move stuff that's inside the nucleus. So I'm just giving you a thumbnail sketch. We're going to go over it in a little bit more detail. I want to give you an overview. So once we get that, and then all the way through anaphase, we start pinching in. So if this were the cell from the side, let me get over here. We just pinch in, and you can see it right there that it looks like it's just squeezing in, and eventually it's going to squeeze in until you have two separate cells. There's some under the microscope. It's pretty cool, but Prophase, visible, we got two chromatids joined at the centromere. Centrosomes are like, well, the centromere is the part that has the kinetochores. It's a region. Centrosomes, and remember those are what the centrioles are, going to opposite sides of the cell we talked about. Now, mitotic spindle we didn't talk about. I glossed over that. Mitotic spindle, those centrioles actually create, remember they're made out of microtubules, they're going to actually grow these, and when they get there, those are going to go. This is how we pull apart the sister chromatids. Those centrioles are going to connect to the kinetochores on opposite, sister chromatids, and at the right time, they're going to start getting shorter, and that's going to pull them apart and move them to the opposite sides where they can form a new nucleus. That's called the mitotic spindle. Asters, or I'll show you that on a picture, it's just more microtubules. Nuclear attached to kinetochore centromeres, the kinetochore microtubules, and draw them to the equator, the middle. Polar microtubules assist in forcing the poles apart. See if we can see that. No, let me get out of the way again. So those are the asters right there. Those are the centrioles, and that is the mitotic spindle. You can see we've got the condensed chromosomes. We can also see that the nucleus is starting to go. The nucleolus is already gone. Okay, metaphase, we got them there, And the reason we put them in the middle is so that we can pull the, I can't see me, so I can't see if it's going right. The reason we put it in the middle is, okay, there we go, is so that they can be pulled apart and go to opposite sides. We just want, if you put them in the middle and you're going to send them to opposite sides, the middle is the best place to start. And there we see them. The mitotic spindle is almost complete. And right there, that's where it connects to the kinetochore. Metaphase is super easy to see. Anaphase, pretty short. That's when these microtubules get shorter and pull the sister chromatids apart. And wouldn't you know it, as soon as they get pulled apart, they're no longer sister chromatids, they're chromosomes. I always thought that was kind of confusing, but nobody asked me my opinion, so there we go. Pulled toward the poles by the proteins of the kinetochores, the microtubules are continuing to force the cell further apart in preparation for cytokinesis. Okay, so there's our anaphase. You can see the microtubules are there, and some of them are still attached, and these guys were never attached to chromosomes. Their job is to try to, lengthen the cell so we can get ready for cytokinesis. Unattached, we already said, chromatids, and they're not chromatids anymore, they're chromosomes. And it depends on who you ask. The terminology is confusing, and I really hate that, but whatever. Okay, telophase, chromosomes are there. They're at the end. And my one from mom and one from dad start to uncoil. We form a new nuclear envelope. The nucleolus comes back, and the mitotic spindle is going to be completely gone. We're just going to disassemble it, and the components are still there. We can reuse it. So there's telophase. You see the mitotic spindle is gone. These guys, they're still chromosomes, and the nucleus is starting to reappear. Now, cytokinesis is the last part of cell division, not a part of mitosis. And we've got microfilaments that just kind of constrict, and they take this big old round middle of the cell and squeeze it until they pinch apart. And again, that's called the cleavage furrow. And we're just doing this to complete the cell division. Meiosis, we're only going to talk a little bit about this. This is something we're going to talk a lot about in AMP2. The main difference between this is that we do 2 rounds. One round is basically mitosis. We're going to just do the same thing we do in mitosis. The second round, we're not going to duplicate our DNA, so when we pull them apart, I only wind up with 1 chromosome. So instead of having 46 chromosomes and 2 sex chromosomes, I have 23 and 1 sex chromosome. That's because when mom and dads come together, then you have the right number. You don't want too many chromosomes. You don't want too few. The N they're talking about is just the number of chromosomes. We normally have two copies. We want to reduce that. We also get something called crossover. Different parts, or mom and dad's chromosome don't necessarily stay intact. They start sharing. A piece of mom's will go over here, and the same piece from dad's will go over there, and that helps us to not make a clone of our mom and dad, but make individual persons. Homologous chromosomes, I should have told you about that. That just means they're the same. I've got one number 1 chromosome from dad and one number 1 from mom. They're homologous in that they are the same chromosome. They're not identical, because your mom and dad aren't identical. Hopefully your mom and dad aren't identical twins. That would be a disaster, and you would probably not be alive anyway. We randomly do this, and this is going to give us the variation. And no 2 cells are going to be alike. They're going to be all different, and different from the one that mom gave. So we're not genetically identical to our mom or our dad because we're kind of mixed in there. So there's meiosis 1. It's just going through there. That's mitosis. This is meiosis 1, which basically is pretty much real identical to this guy. Meiosis 2 is where we go through the division of the DNA, but this time we don't make a copy of it, so we wind up with only half of the normal, which is exactly what we want. I don't want to get too deep into this, guys. I'd rather do this in AMP2. Crossover, chiasmata. Let's see if we got something. Okay, here it is. Here we can see the crossover. So there's mom and dad's. Now this is during meiosis 1, which basically is mitosis, because we're going to double our chromosomes, because we're going to wind up with 4 cells instead of 2 like in mitosis. So there's dad's number 1, there's mom's number 1. What happens is these guys being close together will swap, like I told you, and that way you get some genetic variation. You may have your dad's eyes, you may have your mom's nose or hair or whatever, height, but this is how it goes. So the chromosomes that come out are going to be wildly different. Meiosis 2 we've already talked about, and let's see, that's just a kind of a long drawn out. Well guys, that finishes the cell chapter. I'm glad. I'm pretty sure you are too. It's important. We need this, but it's a lot. But anyway, guys, I will see you in the next video for chapter 4 on the tissues, histology.